Okay, this is our allelopathy group lab report. My name is Alejandra and I'm going to be doing the introduction. And so I found 4 references, and the first reference I'm going to be talking about is in a book. So it's called The History of Allelopathy by R.J. Wills. And so it was able to tell us the beginning and the talk towards the study about the role of allelopathy in the forest. It's known in Australia, and it's under the supervision of Dr. Kinsley Rowan and late Dr. Davin Ashton. And it also tells us what allelopathy is. And so what it is, it's a harmful effect that one plant has on another. And the reason behind this is because there's a chemical that is being released into the environment. Also, a fact about allelopathy is that the word allelopathy was coined in German by Hans Molisch. And they had many interesting facts about allelopathy that I have never known. And then another topic is about allelopathy and invasive species. So I thought this topic was interesting because it's a combination of my other lab presentation. And so it decreases plant growth and soil microbial activity. And from this, what was found is that invasive species depend on biochemical weapons, and that includes allopathic and antimicrobial root exudates. Also, an interesting fact is that invasive species root allelochemicals released into soils. And then another reference was on evolutionary gain. So I thought that one was interesting because it's allelopathy plants release cytotoxic chemicals into the environment. So it increases their ability to compete with surrounding organism for limited resources. And another thing about allelopathy is a chemical, and it's also called allelochemical. So as we know, it's released into the environment and it can negatively affect the growth of a plant. So something interesting is that allelopathy is presented in 72% of the 113 plant families surveyed. So imagine how many that it can have around the world. And then I'm going to be talking about the materials and methods that we use during this lab. So the materials that we needed for the experiment were obviously the plant leaves, the mortar and pestles, and the water, cheesecloth, plastic funnels, the seeds, petri dishes, filter paper, graduated volumetric cylinder, stock bottles, Sharpies. And then, so in activity 1, what we did is we had 3 petri dishes and a Sharpie. And so we marked them 1 water, and the second one is going to be plant 1, so it would be basically plant A, and then plant B. So we remove the dish and then place 1 filter paper, and then we get the seeds and we separate them in 20, and then we do it for all 3 dishes. In activity 2, we went to the garden, clipped about 4 to 5 leaves, maybe a little more just in case. And so in activity...
Hello, my name is Jacqueline Gonzalez, and I will be reading our discussion in conclusion to our lab report. The results did support the hypothesis that the plant would be germinated and that there would be growth. The reason for the results for water is that the radish seeds and water together is what activated the seed growth, because when there's enough water observed, then the outer seed softens and the root emerges from the seed. Then it's followed up with the stem and leaves. But for there to be more growth, then it would have taken a longer time, more than a week. Overall, for plant A and B, there was very small leaves that since we used the juice and poured it into the radish seed, and that activated the growth where there wasn't as much as the one with water. And also maybe because we used different leaves in plant B. In conclusion, what was concluded from the experiment was what caused the growth and how interesting it can be to see the types of different plants and leaves we used, and how to do the data on allelopathy. The results are reasonable because they had growth and it was first on seeing seeds germinate. And the first time it is reasonable because then there is more knowledge about the experiment. There would be more growth for the next one if ever done again. In conclusion, the hypothesis was met that there would be at least some growth, and there were a few of them that were no longer, and some of the seeds were barely growing, but overall it was supported. Thank you. And I will include also the references we used to write the lab report and the reference and proof for going to the library.